For decades, calorie restriction without malnutrition was considered the most reliable way to extend life in various organisms, from single-celled yeast to laboratory rodents. But for a long time, the question remained, does this actually work for long-lived primates and humans? That's why in 1987, the National Institute on Aging launched a study that lasted 23 years. It was conducted on Rhesus macaques, primates that are very similarly to us. As they get older, they go gray. Their hair turns out, body fat redistributes, muscle mass lose tone. And they develop the same chronic conditions: diabetes, cardiovascular disease, and cancer. In this video, we are going to break down the results of the second and longest running study on how calorie restriction affects aging. Hello, my name is George. I'm a biologist with 14 years of experience in fundamental research, and for the last 6 years, I've been working in human clinical trials. Let's dive into the scientific paper together. This was the second major experiment on primates. The paper was published after the results of the University of Wisconsin Medicine study came out, which I covered in my last video. And the goal was the same, to determine if the 30% reduction in calories could slow down aging in animals. The study included 121 monkeys divided into groups based on sex and the age they started the diet. Because scientists suspected that calorie restriction might have different effects depending on whether it starts in young or old age. They split monkeys into two main cohorts. Early onset, monkeys starting diet at a young age from 1 to 14 years old. Late onset, monkeys starting caloric restriction at the older age from 16 to 23 years old. You might wonder why 16 to 23 years is considered old. In the wild, rhesus monkeys usually live up to 23 years, about 3 to 3.5 times less than humans. So this classification makes sense. The caloric restriction group ate 30% less than the control group. Crucially, in this study, the control group didn't have unlimited access to the food. Instead, they received fixed portions twice a day. This prevented the obesity that often occurs when lab animals eat as much as they want. Essentially, the control group ate a healthy norm, while the experimental group ate 30% below that norm. The results were analyzed after 23 years. For the monkeys that started the diet at the age 16 to 23, the survival curves were almost identical. In other words, there was no difference in lifespan between the control and caloric restriction groups in late onset. This suggests that in old age, calorie restriction provides no extra longevity benefit, since the animal is already eating a high-quality diet and not overeating. Strangely, males lived statistically significantly longer than females, 35.4 versus 27.5 years, on average 8 years more than females. This is unusual because in both macaques and humans, females typically live longer. While there isn't a definitive explanation, it is likely a mix of factors. Endometriosis was a common cause of death in the female groups regardless of age, which skewed the data. The scientists didn't provide a clear answer for this phenomenon in the paper. Notably, 4 monkeys from the caloric restriction group and 1 of the control group lived past 40. This is an incredible achievement considering the average lifespan in captivity is 27 years. The researchers believed that top-tier veterinary care, a balanced diet, and avoiding overeating in the control group pushed the animals to the biological limit, a limit that calorie restriction couldn't extend further. In the younger cohort, there was also no significant difference in the survival. By the time of the analysis, 24% of control group and 20% of the caloric restriction group had died, all from age-related causes. However, even though they didn't live longer, the caloric restriction group was healthier. In the caloric restriction group, triglycerides were significantly lower. In the old age, fasting glucose was lower in all caloric restriction monkeys. For old males, this difference was statistically significant. Their glucose levels stayed stable even they aged. At the times, the free radical theory of aging was dominant, so researchers measured plasma-free isoprostane levels. Isoprostane serves as a marker and indicator of a systematic oxidative lipid damage, including damage to the lipids that constitute our cell membranes. Old males in the control groups had much higher levels than those on the diet. This supports the idea that calorie restriction reduces oxidative stress and cell damage. The most impressive results were in cancer prevention. In the early onset cohort, there was no single case of cancer in the caloric restriction group. Meanwhile, the control group had 6 cases, 5 of which were fatal, mostly colon cancer. In the late onset group, the cancer rate was the same for both. This proves that starting early is the key for oncology prevention. As for diabetes, the caloric restriction had 2 cases in the young cohort. In the older cohort, 2 control animals died from diabetes complications, while the caloric restriction group had 0 deaths from it. So while caloric restriction lowers the risk, it doesn't eliminate diabetes entirely. Surprisingly, and contrary to the first study, calorie restriction did not reduce risk of cardiovascular disease. Autopsies show that heart and vascular damage occurred at the same rate in both groups. An analysis was conducted on the time to onset of the first age-related disease, including cardiovascular diseases, cancer, diabetes, or arthritis. In the calorie restriction group, a trend emerged toward an increased age of onset the first age-related condition. This suggests that the quality of life for the monkeys on the diet may have been superior. The results was borderline statistically significant, meaning that the difference between the groups could potentially have occurred by chance. In the results, the NIA study shifted the focus from extending life to improving health. Calorie restriction significantly reduces the risk of diseases, especially cancer, and improves metabolism. Early intervention in adulthood is vital for cancer prevention and immune health. Lower triglycerides and better glucose control are consistent benefits of the 30% caloric restriction in primates. Longevity depends not just on calories but on food quality and feeding schedules. It is still unclear why the 2 major primate studies yield such different results regarding lifespan. I will attempt to explain the reason behind this in my next video, so make sure to subscribe so you don't miss it. If you want to read a full original study, follow the link in the description of video. Please leave your questions, suggestions, or critiques in the comments. I read and answer them all. Thanks for watching.